Good morning, everybody. It's really a pleasure to be here to tell you the details of the Silverback story. Let's start on slide number 4 of the deck, and I'll make a couple of points in addition to what Sonia already mentioned. As we have a very robust discovery and development pipeline that is anchored by our ImmunoTAC platform technology, ImmunoTAC for targeted antibody conjugate. And as was mentioned, it's very versatile, cutting across many therapeutic areas. But the first two programs that we have do target myeloid cells in the tumor microenvironment for localized activation. And this is important because some of these pathways that we're targeting are actually very potent activators of myeloid cells. And by targeting them to the tumor microenvironment, we believe that it provides a product with a greater safety and tolerability profile compared to systemic delivery in an untargeted fashion. Our lead product candidate, SBT6050, is a potent TLR8 agonist conjugated to a HER2 antibody that is in a phase 1 clinical trial. In the first cohort, we have already demonstrated single-agent pharmacological activity. We were able to achieve this because we had a wide therapeutic window in our GLP toxicology studies that allowed us to start at a dose that we predicted to be pharmacologically active, and indeed that turns out to be the case. Importantly, the observations that we're seeing here, increases in cytokines and chemokines that read on myeloid cell activation. You'll also hear that activation of myeloid cells can in turn activate the adaptive immune system, or T cells, and in fact we see other cytokines that demonstrate we've also activated T and NK cells. These are important. Importantly, they read on our other programs, namely SBT6290, which is the same linker payload conjugated to a Nectin-4 antibody with an IND slated for about a year from now. In addition, we see increases in interferon gamma, which are important for our third program, SBT8230, which is the same linker payload conjugated to an ASGR1 antibody for liver-localized activation of myeloid cells for the treatment of chronic HBV. And yes, I'm very honored to lead a stellar management team with incredible experience, and we've already been introduced, so I won't spend any additional time on slide 5. On slide 6 is our board of directors. Many of these names will be familiar to you, and you know them to be people that are very experienced in operating and investing in biotechnology companies. Moving on to slide 7, this is a slide that demonstrates our ImmunoTAC platform, which you can see strategically pairs linker payloads with antibodies. And this does sound like an antibody drug conjugate, and indeed it is, but it differs from traditional ADCs in two important ways. One, we are not delivering the payload to the tumor cell itself, but to an adjacent myeloid cell. And secondly, we're not delivering a cytotoxic, we're delivering an immune modulating agent. And the company has been working on this platform technology for several years, and hence has a library of proprietary molecules that you can see represented on the right-hand table with a series of antigen binding domains and linker payloads. We have a very robust IP portfolio with over 70 patent applications pending worldwide. Indeed, 4 issued patents already directed to our TLR8 franchise. These combination of antigen binding domains and linker payloads has led to a very robust discovery and development pipeline as shown on slide 8. This is our development pipeline here. We are going to spend some additional time talking about the lead program, SBT6050, in a monotherapy dose escalation study, as well as now open for enrollment in a combo with a checkpoint inhibitor. You'll see on the right-hand side of this slide the anticipated milestones, multiple value-generating opportunities for the company. We're going to revisit this later in the presentation, so I won't read these through on this slide. Additionally, as I mentioned, 6290, which is the TLR8 conjugated to a Nectin-4 antibody, slated for an IND a year from now, and right behind that, SBT8230. We also have additional preclinical programs in immuno-oncology with undisclosed antibodies, and then we have a fibrosis program with a TGF-beta receptor agonist conjugated to an ASGR1 antibody for the treatment of fibrosis, and we'll talk briefly about this as well. Let's move on now to slide 9 and dive right in to the lead program, SBT6050. As we talked about, this is a TLR8 agonist conjugated to a HER2 antibody. Importantly, the antibody recognizes the pertuzumab epitope on HER2. This was by design because later in clinical development, we will want to combine this with other HER2-targeted agents, such as Enhertu, Kadcyla, perhaps even Herceptin itself. Each of these are trastuzumab-based, and hence not to cross-block. 6050 recognizes the pertuzumab epitope. We'll walk you through 6050's differentiated profile. It's been well tolerated in the ongoing phase 1 dose escalation study with no dose-limiting toxicities observed to date. We've enrolled 6 patients at the 0.3 mg per kg dose of 6050, and we've learned an incredible amount. We'll show you some of those data. The second cohort is now open to enrollment. As of last week, we've had 1 patient that's been on study for 17 weeks. So already we've learned a lot about the repeat dose safety and tolerability. Importantly, pharmacological activity has been observed in patients. We've seen increases in chemokines and cytokines that indicate we've activated myeloid cells. Myeloid cells in and of itself have anti-tumor properties, and we activate T and NK cells, and this is as a result of myeloid cell activation, which then in turn activate T and NK cells. This is important for durability of response. In these 6 patients that have been treated, we've had 2 patients that have had radiological reassessment, 1 patient at 8 and 16 weeks who had stable disease, and another patient at 8 weeks which showed a greater than 30% reduction in the target lesion diameter. Moving on to slide 10 now, we're going to get into the details about TLR8, the preclinical data with 6050, and then moving into the clinical data. I'd like to turn it over to Dr. Valerie Odegaard now to continue. Valerie?
Thanks, Laura, and good morning, everyone. When we initiated this program, we selected TLR8 because of its expression profile in human myeloid cells. As you can see in slide 10, TLR8 is robustly expressed in myeloid cells such as macrophages that, when activated, can directly kill tumor cells. And it's also expressed in cell types such as conventional dendritic cells that, when activated through TLR8, promote T cell responses as well as amplify NK and B cell responses. Importantly, TLR8 is only expressed within the myeloid cell lineage in humans. It's not found outside of that lineage, resulting then in restricted activation of those cell types. On slide 11, you'll note that there are others in this space in terms of TLR agonist conjugates. Importantly, SBT6050 is differentiated in a couple of key important ways. The first is the selection of the payload. 6050 again incorporates a selective TLR8 agonist. In the prior slide, we touched on why we think that TLR8 is an important target for activating myeloid cells. Others in the space are focusing on TLR7, either with a selective TLR7 agonist or with a resiquimod-like variant, which is heavily skewed towards TLR7 activation. TLR7, unlike TLR8, is expressed primarily in humans in B cells, plasmacytoid DCs, and outside of the immune compartment. We know in terms of status that the Novartis program, as shown in the center of the column, has been halted. They recently gave an update this year at SITC, and Bolt Biotherapeutics is in a phase 1/2 dose escalation and has not yet disclosed data from their program. The other important point I'd like to note in terms of differentiation comes back to the HER2 binding domain. Again, as Laura already highlighted, SBT6050 incorporates a binding domain that recognizes the pertuzumab epitope, which allows for combination, again, later in clinical development with trastuzumab and trastuzumab-based agents such as in HER2. And again, this is different from others in the space. On slide 12, then, you can see the mechanism of action in terms of delivery of the conjugate into myeloid cells. You'll see on the left-hand side of this slide a cartoon depiction of SBT6050. SBT6050 is systemically delivered, administered through subcutaneous injection, and then it's taken up by myeloid cells via Fc gamma receptors expressed on those myeloid cells. Importantly, that Fc gamma receptor uptake is mediated by binding to HER2 on the tumor cell at the same time. This coincident binding with HER2 then results in receptor internalization of the Fc gamma receptor and delivers the conjugate directly into the endolysosome, which is where TLR8 resides, resulting in an activation of myeloid cells in a HER2-dependent manner. Importantly, SBT6050 has been optimized to be equipotent in the setting of both high HER2 expression, that which is defined as 3+ by IHC, but also what we refer to as a moderate level of HER2 expression. That's a HER2 expression level of 2+ by IHC, even in settings of FISH negativity, again, because this mechanism of action does not require HER2 to be an oncogenic driver in order to mediate its myeloid cell activation and anti-tumor effects. Slide 13 introduces you to some of our human ex vivo data. What you see on this slide is data from a co-culture assay in which human immune cells were co-cultured with HER2-expressing tumor cells in the presence of SBT6050. As you see indicated on the purple lines on the graph, 6050 induces robust activation of myeloid cells in a HER2-dependent manner and does so through a number of different responses. For example, production of tumor necrosis factor alpha in the top left graph. This is a mediator that directly results in the killing of tumor cells. IL-12, for example, from dendritic cells, in turn primes and amplifies T cell and NK cell responses. And you can see that even in these human ex vivo assays where there's robust induction of interferon gamma and granzyme B, the key effector mediators from T cell and NK cells. Importantly, across all of these different mechanisms, SBT6050 is potent, again, in the presence of HER2 with an EC50 of about 200 picomolar. If we look then at slide 14, you can see that the activity of 6050 with a TLR8 selective agonist cannot be recapitulated with conjugates in which a TLR7 agonist or a resiquimod variant payload are used. And you can see that in the head-to-head studies on the right-hand side of the page. Importantly, this comes back to the differential expression of TLR8 in human myeloid cells as compared to TLR7, which again is primarily expressed in B cells and plasmacytoid DCs, and hence you see that conjugates with TLR7-based payloads just don't have the same functional profile as SBT6050 does on myeloid cells. If we move then to slide 15, we'll look at some of our preclinical in vivo studies. For these models, we generated a surrogate that is designed to recapitulate the activity of 6050 on human myeloid cells, but of course in this case, for the surrogate, it has the same activity on mouse myeloid cells. This required an adjustment to the Fc binding domain to allow for engagement with mouse Fc gamma receptors, and also use of a TLR7 agonist payload in mice, because in mice, TLR7 as opposed to TLR8 is what's expressed robustly in those myeloid cells. This surrogate has the same potency as SBT6050 does on human cells. What you see then on page 15 is that the SBT6050 surrogate has robust single-agent activity, even in mouse models as shown on the left-hand side that lack T cells and NK cells, but only have an intact myeloid cell compartment within the immune system. You can see, again, robust tumor cell clearance in that setting, indicating that myeloid cells, when appropriately activated, can directly kill tumor cells. In the middle panel, this is coming back to a different mouse model, what we refer to as a syngeneic model, one in which all of the various immune cell types are present. Here again, in this model, you can see robust single-agent activity. And in fact, when we come back and re-challenge these animals, which do have T cells, with tumor cells months after their initial clearance of their primary tumor, with the addition of no additional drug, they're fully protected from this tumor re-challenge. You can see this in the far right panel, indicating that the activation of dendritic cells in these syngeneic mouse models then in turn prime potent memory T cell responses against the tumor. And those memory T cell responses are no longer restricted to HER2. They're broad in terms of their tumor reactivity and specificity. As we look then at slide 15, the take-home message on this slide is that in our GLP toxicology studies, SBT6050 demonstrated a broad therapeutic window, which then enabled us to start at a clinical dose that we projected to be active in terms of pharmacologic activity. We've now shown that this is the case, as Laura already referenced in her introductory remarks. To go through this slide in a bit more detail then, first in the table, you'll note in the top row the dose levels that were evaluated in our NHP studies. Again, because of the tolerability of SBT6050 across multiple studies, we recommended to the FDA, and the FDA agreed with our recommendation that our starting dose for 6050 in the clinic should be based on the minimum pharmacologically active dose in the monkey, that was 0.5 mg per kg, and then scaled then to a clinical starting dose of 0.3 milligrams per kilogram. This starting dose for an immune agonist at a pharmacologically active dose level is not typical. Typically, novel immune agonists start at, for example, a dose defined by the MABEL, resulting in a subtherapeutic dose level, which requires then a long dose escalation in terms of then reaching dose levels projected to have clinical activity. Here with 6050, because of the starting dose, you can see, looking at the bottom row of the table, that 0.3 milligrams per kilogram is close to dose levels where we project clinical activity could be observed, again, based on the modeling from our preclinical data. What you see then on the graph at the bottom are a couple of representative examples from our first patient. And Naomi, as she'll walk through our clinical experience, will get into more detail around our clinical experience to date. But in these graphs, what you see on the left-hand graph is that for the first patient, looking at their biomarkers post their first dose, is that the pharmacologic activity, just using IP10 as one example, is exactly as predicted based on our preclinical studies. And furthermore, that the elevations and induction of pharmacologic activity is maintained with subsequent doses, and you can see that on the right-hand graph. And so with that, then, I'll turn it over to Naomi, who will talk through our clinical experience with 6050 to date. Naomi?
Thank you, Valerie. So I'll start on slide 17 with a snapshot of our current progress in the clinical trial. So in the single-agent dose escalation, we are enrolling patients with HER2-expressing solid tumors, which can be 2+ or 3+ expression by IHC or HER2 gene amplification. In the first 6 patients, we've enrolled a wide range of tumors, 2 breast, 2 non-small cell lung, 1 gallbladder, and 1 esophageal cancer patient. And some have HER2 amplification and some are not amplified. SBT6050 is given every 2 weeks by subcutaneous injection, and formal tumor reassessment scans are done every 8 weeks. As of November 25th, we've dosed 6 patients at the 0.3 mg per kg level and have completed the DLT observation period for that dose level. Overall, therapy has been well tolerated with common adverse events, including mild to moderate fevers, chills, nausea and vomiting, and redness or swelling at the injection site. So these are not unexpected for an active immunotherapy agent. ADA assessments have shown low titer antibodies in some patients, however, no impact on the pharmacodynamic activity. 2 patients have been on treatment through multiple cycles and had formal tumor reassessments, and we'll discuss those 2 patients in detail in the next few slides. So going to slide 18, the first patient enrolled has now been on study treatment for 17 weeks. She's a 73-year-old breast cancer patient who was initially diagnosed with early-stage disease and was treated with neoadjuvant therapy, surgery, radiation, and maintenance Herceptin. Since her recurrence with metastatic disease, she's received multiple lines of HER2-directed therapy, including Kadcyla, ZW25, and PRS-343. At the time of enrollment, she had actively enlarging pulmonary nodules. She has now received 9 doses. With the first dose, she had moderate fevers, chills, and injection site reaction. However, with subsequent doses, she's managing these very well with pre-medications. In her peripheral blood, we see elevations of IP10, MCP1, and IL-6, demonstrating myeloid cell activation right in the range predicted by NHP studies, as Valerie showed you. Importantly, we are also seeing interferon gamma elevation, indicating that secondary activation of NK and T lymphocytes is occurring. And we are seeing these elevations continue after each repeated dose. Her 8 and 16-week tumor reassessments show a stabilization of previously progressing pulmonary nodules. On slide 19, we're also seeing transient changes in hemoglobin, exactly as predicted by the NHP studies. These decreases are predictable, dose-related, and recover prior to repeat dosing at 2 weeks. These changes can be viewed as a pharmacodynamic marker, and we believe the decrease is due to TLR8 activation and downregulation of SIRP-alpha on macrophages, blocking the don't eat me signal. On slide 20, the third patient enrolled has now been on study for 10 weeks. She's a 66-year-old woman, never a smoker, with HER2 2+ expressing non-small cell lung cancer with brain metastases. She was previously treated with chemotherapy and nivolumab and radiation. However, she recurred in early 2020 with metastases in the brain and chest. The brain met was treated with radiation again, and nivolumab was reinitiated at the beginning of May. The CT scan in June showed stable disease. However, by August, she had new progressive mediastinal lymphadenopathy in the chest. These 5 doses had toxicities that are quite manageable, and like for the first patient, we are seeing the cytokine elevations after each dose, including the interferon gamma response. And then on her 8-week CT scan, she had significant improvement in the mediastinal lymphadenopathy with a greater than 30% decrease in target lesion diameter, which meets the RECIST criteria for an unconfirmed partial response. So going to slide 21, just wrapping the clinical observations, we've completed the first dose level. There are no dose-limiting toxicities. It's still very early data. However, this kind of data to see in the first cohort is very encouraging. The safety profile is consistent with an active IO agent. We have manageable flu-like symptoms and injection site reactions. 2 patients have been on treatment for many months, and we're observing cytokine elevations as predicted by the NHP studies that support our proposed mechanism of action, suggesting not only myeloid cell activation, but also evidence of lymphocyte activation. In the first 2 patients who have had disease reassessment, We see stabilization of previously progressive disease in one and tumor regression in the other. The investigators on the study have been very enthusiastic, and several patients are lined up to start treatment in the next cohort. Going to slide 22, we are now recruiting for dose level 2. When the RP2D is determined, we'll open expansion monotherapy cohorts, which have been planned in settings of high unmet need that would be appropriate for accelerated approval approaches in the future. Also, we have met our predefined threshold for opening the pembrolizumab combination, and the first dose level is open for enrollment. Because both of these are IO agents, we've opted to do a parallel dose escalation with our single agent therapy. The additional combos that we are looking to explore when we determine the RP2D include trastuzumab-based therapies like Enhertu and also chemotherapy plus trastuzumab. I'm going to slide 23. So HER2-directed therapies are the foundation of therapy in breast and gastric cancer that test positive for HER2. However, HER2 expression and amplification are being increasingly recognized as an important target across common aggressive tumors that have high unmet need. And these provide multiple opportunities for accelerated approval approaches and also combinations with checkpoint inhibitors and standard therapy with or without trastuzumab. Slide 24. Our initial focus is on fast-to-market opportunities in relapsed or refractory disease, but at the same time, we're laying the groundwork for combinations in earlier lines of therapy. The characteristics of SBT6050 make it an ideal agent for novel combinations in a curative setting where we can make the maximum impact for patients. Priority combinations will include trastuzumab-based therapies such as Enhertu and also checkpoint inhibitors in earlier lines of therapy. So I'll hand off to Valerie now to discuss some of our earlier agents. You can see why we're encouraged so far with what we're seeing in the clinical trial and are looking forward to seeing if we can extend these observations to additional targets.
Thank you, Naomi. So if we turn our attention then to page 25, we'll give you a brief introduction to SBT6290. This is the second program in our portfolio and leverages the same TLR8 linker payload that you've just heard about in the context of 6050, but now conjugated to an antibody directed to Nectin-4. Nectin-4 is a clinically validated target through the approval of PADCEV in bladder cancer. And in fact, SBT6290 has now demonstrated the same functional profile as 6050 in ex vivo human assays, and even in non-GLP toxicology studies has also been supportive of a wide therapeutic window for this agent. This program is on track for IND submission in the fourth quarter of next year. On slide 26, you see brief snippets of data supporting SBT6290. The first on the left-hand side is that this agent, again, in this case in a Nectin-4 dependent manner, potently activates myeloid cells, again with a potency that is essentially identical to that of SBT6050, highlighting the transferability of this TLR8 linker payload across antibodies that recognize diverse target antigens. On the right-hand side is data to support that SBT6290 blocks the interaction that has recently been described between Nectin-4 and TIGIT. TIGIT, of course, is an immune checkpoint molecule, and Nectin-4 has recently been described to be a ligand for that receptor. The binding domain of 6290 can block that interaction and hence then adds an additional mechanism by which this agent could amplify T-cell and NK cell responses. On slide 27, with a surrogate of 6290 in mouse models, again, a very clear demonstration of single-agent anti-tumor activity. And similar to the markers that we've shown you with 6050 in the first cohort of patients, we see markers of myeloid cell activation and markers of T-cell activation in mouse tumors that are responding to 6290 surrogate across these mouse studies. On slide 28, then, we are continuing to expand our TLR8 IO franchise. There are two additional programs that at this point are undisclosed that again leverage the same TLR8 agonist linker payload and to date display the same profile that we've just disclosed and discussed on 6050 and 6290. With that, then, we'll turn our attention to slide 29, which introduces our third program, SBT 8230. In this case, the TLR8 agonist payload and linker, again, that have just been described in the context of 6050 and 6290, is conjugated to an antibody directed to ASGR1. This conjugate then allows for liver-localized activation of myeloid cells, and this is an agent that we are developing for chronic HBV. This program is moving towards IND submission with a date of second half of 2022. Importantly, the data that has been generated with 6050 in the first cohort of patients further supports the advancement of 8230. In the context of chronic HBV, the generation of interferon gamma-mediated immune responses has been shown to be associated with achievement of functional cures. And of course, that is the mechanism that is induced by TLR8 agonism, again, as you've seen even in the first cohort of patients with 6050. It's because of this link between TLR8 biology and the generation of these interferon gamma-mediated immune responses and seroconversion in the context of chronic HBV that has enabled and provided the rationale for others to pursue agonism of TLR8 as a potential approach for treatment of chronic HBV. Gilead has an untargeted, orally administered TLR8 agonist, but again, this results in the activation of myeloid cells outside of the liver that can be dose-limiting. 8230, again, results in liver-localized myeloid cell activation, binds to ASGR1, but in a manner that allows for a combination with RNAi approaches that use GalNAc, which also binds to ASGR1. So these agents are not cross-blocking. Again, development candidate has been selected based on a series of preclinical studies, both ex vivo as well as initial exploratory NHP toxicology studies as well that demonstrate a wide therapeutic window. On page 30, you'll see some data using a surrogate of 8230 in a mouse model of chronic HBV. Importantly, this is a model where it has been very challenging to demonstrate seroconversion because of the immunosuppressive nature of this model, really replicating what's been observed in human disease. Here, of course, as you see on the right-hand side, the surrogate robustly induces seroconversion, and of course, this is associated with reduction in hepatitis B surface antigen plasma levels. In addition, not shown on this slide is that the surrogate also lowers viral DNA titers, as you would anticipate. And finally, we'd like to introduce you to our fibrosis program. The Immunotech platform, importantly, is not limited to the delivery of TLR8 agonists or agonists generally. And in the case of the fibrosis program, in fact, we are leveraging this technology to deliver proprietary TGF-beta receptor antagonists, in this case, conjugated to an antibody targeting ASGR1, the same antibody we just introduced you to in our HBV program. And this conjugate then is designed for the treatment of liver fibrosis. Currently, this program is in preclinical development, and look forward to advancing this program and providing more detail in short order. And with that, I'll turn it over to Jonathan, who will cover the financial highlights.
Thank you, Valerie, and good morning, everyone. So the company has raised $218 million to date, $142 million of cash on the balance sheet as of the end of September, and the company has cash runway of at least the next 24 months pro forma for this IPO. On the right hand of slide 32, you can see the three financings that the company has done and the milestones that have been achieved that the company has, that we've spoken about just now. So next slide, we see a number of value-generating milestones over the next 6 to 18 months. I'd point you to the 6050 monotherapy study where we'll see interim phase 1 data in the second half of 21 and additional data in the second half of 2022. 6050 combination data will come in the first half of 2022. 6290, the Nectin-4 program, will submit an IND in the fourth quarter of 21 and initiate phase 1 in the first quarter of 22. And our HBV program will submit an IND in the second half of 2022. So a lot for us to be very excited about. So slide 34, I'll wrap here with our key investment highlights. Our mission at Silverback is to achieve victory for patients. And we've got a really versatile platform in the Immunotech platform to unlock a new class of targeted IO agents. Really, we're waking up the immune system by activating and reprogramming myeloid cells. 6050, our lead candidate, has single-agent pharmacologic activity observed in the clinic at the first dose level. We're very excited about our progress there. 6050 really does unlock the potential of the TLR8 franchise, not just in IO, but looking at virology and beyond. And as I described, a lot to be excited about in the next 16 to 18 months with the number of interesting milestones, and importantly, a proven management team that has the ability to shepherd this important technology through the clinic and to patients. And with that, I want to thank you for your time and attention,